And a Christmas tree in Canada and in Bordeaux looks the same, okay? It's just a Christmas tree. It's a pine tree. But what makes the difference is the modifications that you add to this Christmas tree. And that is the decorations that you put on this Christmas tree will now make it different from one household to another. As you see also in the beautiful decoration of the orchid, of the Easter orchid, that is also quite different from the other orchids. So our objective today is to try to explain to you how post-translation modifications actually change the properties of the protein and give it multiple functions. So many years ago, for a number of years, we have observed that many proteins are actually synthesized as what is known as precursors. In other words, they are big proteins that need to be cut into smaller proteins to release an active fragment. It could be a growth factor, a receptor, a pathogenic protein, and even transcription factors like Notch and so on. And it was noticed that all of them have one thing in common, and that they are cleaved at single or paired basic amino acids. And we came up with a motif that is common to all of them, some kind of, and that motif suggested that there must be an enzyme or a series of enzymes that recognize such a motif so as to generate the diversity of functions that we observe. And it took us, that was an easy question, very difficult to answer. It took us more than 15 years to identify the enzymes responsible for such cleavage of these secretory proteins. And you know, the genome from Handel and so on, Mendel, it started a long time ago, but really the genome project started in 1990. So the first hint of a convertase that could transform an inactive protein to an active one came from yeast, from alpha-mating factor cleavage. Then a second one came from an insulinoma by John Hutton in England, who discovered that insulin may require the activity of two enzymes to make it. And when the genome project started, I joined the Pasteur Institute at that time in France, and I began to actually try to investigate what are the enzymes responsible for this process. And it took a number of years, and you can see that over the years, we were able to identify the 9 enzymes involved in this process, and each one of them has a different function, as you can see. And I will not be able to cover all the functions because it's just a big project, but I will cover some of them and their relationship to pathology. So this is how they look like. Took a long time, you know, I was having a lot of discussions with the biggest enzymologists in the world as to decide what kind of enzymes these are going to be. They turned out to be serine proteases related to subtilizing, to bacterial subtilizing. That's a very old project. In other words, this is not a new discovery. They're actually bacterial origin, and they were modified to generate the proprotein. And there are 9 of them. 7 of them cleave at those pair basic amino acids, like I told you. It's called PC1, PC2, furin, PC5, phosphor, and PC7. And there are 2 of them that do not cleave at basic residues, but cleave at non-basic residues, very important for regulation of cholesterol and fatty acids. And this one, the last one, is what you've heard from Majid just told you, is now in clinics. Antibodies against this one are to diminish cholesterol. Now, all these enzymes are synthesized in the same way. They're all of them activated in the endoplasmic reticulum of the cell to remove their prodomain, which is a chaperone. And once the chaperone is cleaved, they still remain inactive. They need to exit the ER, move to a compartment whereby a second cleavage or a separation occurs such that you have a catalytic activity. So we began to use a lot of electron microscopy and a lot of analysis of cells to identify where they're localized, these enzymes. And you can see that it's quite diverse. They are localized in the cis-medial Golgi, in the trans-Golgi network at the cell surface, and they can cycle back to the Golgi, or they enter secretory granules such as PC1 and PC2 and make all the hormones that we know. And some of them are actually secreted, such as PC5 and PACE4, as well as PCSK9, which is very unusual, as you will see. It is secreted with its inhibitory prodomain. Now, in general, what we found is that any secretory protein that enters the secretory pathway that needs to be cleaved, and we estimate around 3,000 of the genome proteins that are going to be cleaved like that, either is cleaved to generate a new function, such as what we call an activation, a gain of function, such as the creation of insulin from proinsulin, for example, or they can actually go the other way. It can actually be inactivated by cleavage. So in other words, cleavage can generate multiple functions, which will give us this diversity that we're looking for. So over the years, a number of people, including ourselves, identified a number of substrates which are cleaved by these enzymes. And you can see that PC1 activates ACTH glucagon-like peptide. PC2 makes beta-endorphin and glucagon. Furin makes a large number of proteins, including growth factors. We talk about them now, Notch and TGF-beta, integrins, alpha-V, even viral glycoproteins, such as HIV cleavage and bacterial toxin. And each of the other ones have different substrates, as you can see. PC7 seems to have a very limited number of substrates, such as transferrin receptor. And finally, those two proteases that we know very little about still, they're non-cleaved basic residues, but they're involved in the activation of membrane-bound transcription factors, which regulate a large program in the cell in terms of fatty acid metabolism. And also they regulate the production of lysosomes. And they can activate some viral glycoproteins. And I'm going to finish with PCSK9, which is actually a chaperone targeting certain receptors to degradation. So here's the first example. Here is a precursor known as pro-opiomelanocortin. It is the precursor of ACTH and beta-endorphin, and another precursor called proinsulin, which is the precursor of insulin. In other words, these proteins start as inactive proteins because they are involved, they are attached to these prodomains. And then comes PC1, the first enzyme that we identified. It cleaves certain dibasic residues to release ACTH, as you can see. But it also cleaves insulin only on one side. So PC1 is not enough to make active insulin. You need a second enzyme. That's what was discovered in the insulinoma. I just told you from John Hutton that he needs two enzymes to do that. It turned out that to be the PC2 with its chaperone 7B2 will then cut it back now to release beta-endorphin and alpha-MSH, as well as active insulin. Now, why is that important? Then, of course, you have a carboxypeptidase to remove the dibasic residues that are now exposed. Why is that important? It turns out that there is a disease, a genetic disease called the Prader-Willi syndrome. It affects 1 in 125,000 live births. Now, these kids are fantastic. I've seen them, I've met them. They're laughing all the time, but they're extremely heavy, as you can see. They are also, if you want, they have gonadal problems. They are diabetic. They have a lot of endocrine diseases. And it turned out that this disease is a genetic deletion of a non-coding RNA called SNORD116, which codes for a helix-loop helix transcription factor, which then codes for PC1. So if you lose this non-coding RNA, you have much less PC1, and that has repercussions. You have much less ghrelin, you eat much more, you have much less growth hormone because of less of growth hormone release. In fact, that's why they are small. You have diabetic because you have less insulin, and so on. And it's a paternally expressed gene. So just the loss of one of the enzymes, you can see the amount of tremendous problems that you can get. So of course, to understand better the function of these enzymes, we had to go to a much longer process, which is the identification of the in vivo function of each one of these enzymes. And it took us a large number of years. I can tell you that we've now exceeded 120,000 mice in the lab to be able to identify some of the roles of these convertases in vivo. And we've generated knockouts of each one of them. And they are diverse. The PC1, of course, they are dwarf, and they have problems of insulin, as well as PC2. Furin, they die, okay? At embryonic day 11, they have problems of axial rotation and no heart looping. PC4 are infertile. PC5 have problems of Hind lipoplasia. PACE4, they have cardiac problems. And PC7, amazingly, they're alive and they're very happy and they're less anxious. And SCI1, actually, they die at embryonic day 3.5. And finally, the last one, they're alive also, and they are very low cholesterol. So really, two only of the convertases, when you remove them, the animals are very happy. In fact, we've now done a double knockout of both of them, and they're also very well, and they have a lot of advantages. So if you look at the diseases that affect humanity over the years, there were a number of diseases at the beginning of the 20th century. You can see that tuberculosis, neuropathies, and so on. But as the antibiotics have been developed and other diseases have been solved, the major diseases that remained are cancer and heart disease. And now, if you look at even now, the amount of people dying from heart disease or cancer is almost equivalent, as opposed to all the other diseases which have been relatively better controlled. And now with the COVID advance, it became the third leading cause of death in the United States, as well as in many other countries. And it's about half of what are dying from heart disease and cancer so far. And so today, what I'm going to discuss with you is the role of the proprotein convertases. We'll start with the viral infections, which is, if you want, so far, the one that causes less death than the other two, move to cancer, go to cardiovascular diseases, and then back to viral infection. And you can see how there is a loop, that the enzymes involved in all these processes are actually connected. So let's just say, what do these convertases do? As you can see, that PC1 is involved in obesity and diabetes, PC4 in fertility, but I'm going to be concentrating today on three of them: furin for cancer and viral infections, PC7 for cancer and triglyceride and ApoB regulation, and finally PCSK9 in cholesterol regulation and viral infections. So let's start with furin. So what is furin doing? Furin is everywhere. It's expressed in every cell. It is affecting a large number of substrates, as I told you, the mammalian substrates involved in cancer, diabetes, inflammation, and neurological diseases. It involved in the viral substrates, such as the HIV, the Bunya virus, the Toga virus, the flavivirus, many kinds of viruses. It activates the cell surface glycoproteins of these viruses. And even bacterial substrates, such as anthrax toxin, diphtheria toxin, and so on, and it affects bacterial infections. So obviously, when you see an enzyme like this and you ask the question, well, wait a minute, does the cell develop a method to regulate its activity? Is there an endogenous inhibitor of furin? So we looked at the role of furin in multiple viral infections, and you can see that it's always the same. It cuts the viral glycoprotein at pairs of basic amino acids. You can see it here. Even in Hong Kong, they had to kill 1 million chicken because there was an insertion of 4 amino acids in front of the cleavage site that made it such an efficient cleavage site that it was extremely rapidly spread. It reminds us a little bit of what's going on now with the COVID, these variants. And now we can see that even the Sars Coronavirus also has a single basic cleavage site, which I'm going to show you is done by furin. Now, looking for natural inhibitors, we used HIV patients and the brain of HIV patients to identify that actually there is inflammation of the brain of HIV patients, and that the body reacts by overexpressing a protein called protease-activated receptor 1. It's a 7-transmembrane receptor, which is upregulated together with furin in the brain of HIV patients. Now, why is that? It turns out that it doesn't actually interact with the catalytic subunit of furin, as you can see here, but rather it blocks furin and itself in the trans-Golgi network. In other words, normally the PAR1 is at the cell surface, but in the presence of furin, it is blocked at the trans-Golgi network. And that is because in this loop, there is a sequence recognized by furin, and the two of them block each other. So that was the first natural inhibitor of furin that we identified. And it's not an inhibitor of activity per se, it's an inhibitor by blocking it at a given site in the cell. A German group recently also identified another inhibitor called the guanylate binding proteins 2 and 5. And that is induced whenever you have a viral infection, the body tries to react by synthesizing cytokines such as interferon gamma, for example. And this protein is induced by interferon gamma. And it also blocks furin by interacting with the cytosolic tail now. This is a cytosolic protein and blocks it in the cis-Golgi in another way to block the activity. And of course, we wanted to ask the question, is this going to happen also with SARS coronavirus? So talking about SARS coronavirus, it reminded us of pandemics. Pandemics are not new. They started around 12,000 years ago when the man started, instead of being a hunter-gatherer, began to form groups in villages and so on, and began to plant and live out of plantations. So the first plague that we know is known as Antonine plague in ancient Rome, and it happened in about 165 or 180 after Jesus Christ's birth. 5 million people died there, okay? So there are a number of plagues that happen over the years. As you can see, the Black Bubonic Plague was a huge one. It killed 200 million people in the 14th century. You've got the Spanish flu in the early 20th century in the Second World War that killed about 50 million people, as well as the HIV/AIDS now that we live with. There is about 35 million people died, and now come the COVID, okay? Now, it turns out that COVID is a coronavirus, and it's actually the first, it's related to a coronavirus that was identified in the early 1900s or at the end of the 19th century in Russia. And it was known as the OC43, which is an endemic virus right now. So really it is a cousin of an already known virus that is endemic in our societies. So coronaviruses really, we identified by June Almeida when she looked at the electron microscope in the early 60s of a sample of a virus, and she found something funny about it. She said, well, there's some kind of a crown on top of it, as she observed it in the electron microscope. And that's why it's called coronavirus, because it looks like a crown. And really 4 of them, the OC43 I just told you about in Russia, and 3 others have been identified, and they are now endemic. They run in our society, and every year 20% of the flu viruses are due to them. However, really, in 2002, 2003, Canada has suffered a lot from that. Was the first SARS coronavirus, we call it SARS-1. These are beta coronaviruses, followed by a Middle East coronavirus that infects the camels and dromedaries in 2012, and finally the 2019, the SARS coronavirus 2. These are the human pathogenic viruses that we live with. So when the sequence of the SARS coronavirus came out in the early January of 2020, rapidly, we were able to look at the sequence and realize that there is an insertion of 4 amino acids ahead of the, close to the site that is known to be cleaved for the spike glycoprotein. And now that created a furin site. It became RXXR. So we suggested that furin may be involved in the activation of SARS coronavirus. And then there is a second cleavage called S2', which was found also in SARS-CoV-1. So the difference between SARS-CoV-1 and SARS-CoV-2 is really in the S1, S2 site because of this 4 amino acid insertion. And so the question we asked, like many other people are trying to ask whether we can target a number of these proteins from the virus, such as the helicase, the 3CL protease, and so on. We're asking the question whether targeting furin could prevent entry of the virus inside the cell. Now, if you look at the way the viruses enter cells, it's like a key entering a keyhole. The key is the spike, is the surface glycoprotein. The keyhole is a receptor, and that is known as angiotensin converting enzyme 2. It's not acting as an enzyme, it's acting as a receptor here. So once these two meet, you get fusion. How does that work? Well, it turns out that the fusogenic sequence, this is the spike glycoprotein, has an N-terminal big part here, it's called the cap, and a C-terminal part, which is the fusogenic sequence. Once it binds the receptors in the conformation at which it is in an up form, it will separate away after cleavage, but what we think was furin, and that exposes the second cleavage site called S2', that allows fusion and delivery of cargo. And so the question is, the whole game is to detach the cap from the C-terminal domain, and that requires proteases. And our job is to identify which proteases are doing this. So we first started about almost a year ago now by making a codon optimized cDNA for code coding for the spike glycoprotein, added the V5 tag, and express it in cells. to our delight, we found that if you express it into the cell, that pro spike is cleaved by endogenous, what we now know is furin, into the exactly at the S1, S2 site. We've done the proteomics and we've proven it is the site. Now, we also saw a small amount of cleavage at what we thought was the S2 prime site, and we just had the proof now by proteomics that it is. If you mutate the arginines at P1 and P4, you completely lose the furin cleavage. So that confirms that it is the site. If, however, you overexpress furin, you get a bit more of the S2 prime. Interestingly, the TMPRSS2 is a transmembrane protease reported to be important for SARS-CoV-1. And people said that it may play a role in SARS-CoV-2. And so we wanted to co-express TMPR with the wild type S. And to our surprise, we did not. First of all, we lost the furin cleavage. And we generated two fragments which have nothing to do with the two fragments that we saw with furin. So what's going on? And that these two fragments are generated even in the mutant of the two arginines here. But notice that we've lost the furin site. So this suggested that TMPRSS2 must be acting before furin can cleave. And when we add the receptor called ACE2, you get enhanced cleavage at S2 prime, which means more infections, and also enhanced cleavage at the lower band here called S2B. We had to solve this conundrum and understand what is TMPRSS2 doing. So we synthesized peptides, mimicking, if you want, the SARS-CoV-2 sites that we predicted. And in fact, S1, S2, as well as S2 prime are cleaved by TMPRSS2. So what are we observing? How come we're getting different sizes of products? Well, we solved that problem, finally. We had to use cells that do not make O-glycosidation. And we found out that the enzyme TMPRSS2 can auto-activate itself in the endoplasmic reticulum and then cleave pro-S in the ER into S2A, which turned out to be S2 by furin, and S2B is nothing but S2 prime. So in other words, these two products have a different size because they're not O-glycosidated. But this will kill the S. So how come TMPRSS2 is an activator? So for this, we had to develop what is known as a co-culture. We mimic the story of the virus. So basically, we come with cells which express TAT, which is a protein isolated from HIV, and add and express S, and co-culture it with another cell under the control. Well, luciferase is under the control of a TAT promoter. And so when the two proteins meet, the S meets its receptor, they will fuse. They will transfer TAT from the donor to the acceptor cells, and you get chemiluminescence. So fusion is detected by this Phytogenic assay. And we found out that if you do this co-culture now by adding TMPRSS2 in the acceptor cells and S in the donor cells, yes, indeed, TMPRSS2 can cleave both the natural S as well as the mutant, as we saw, S1, S2. And that was a surprise. But if you look at the sequence of the mutant that we generated, we replaced arginines here by ALA, but we still left one arginine. And so it's still cleaved at the AR. That explains it. And now we have a triple mutant and it's no longer cleaved. So we can conclude that both TMPRSS2 and furin cleave what we believe to be at the same sites. Now, how do we do with furin? We had to develop specific inhibitors of furin, and we need a non-toxic, non-peptidic inhibitor of furin. And we collaborated with a company called Boston Pharmaceuticals, and we have screened a large number of compounds, of which 3 of them were identified. As you can see, their structures here. One by using a fluorogenic, if you want, assay in vitro. And you can see that these inhibit these 3 molecules, BOS 318, 981, 857, inhibit as well as the RVKR chloromethyl ketone. And these are the number of experiments we've done. So it's really reproducible at around the 10 nanomolar range. We then developed a Golgi assay, whereby we attach a sequence from the bone morphogenic protein to GalNAc-T2, and it's blocked in the Golgi. And now you see if the enzymes, endogenous enzymes, are blocked by this, and it did, by releasing of the GFP fluorescence. And finally, we did a synthetic peptide that mimics the Sars Coronavirus and found out that 981 inhibits the cleavage by furin of the Sars Coronavirus at around 4 nanomolar IC50. But what's important is in cells. Do these inhibitors actually enter cells and inhibit the endogenous furin? The answer is yes. You can see that both of the three of them inhibits the processing and the S2 prime as well as S2. So now we were ready to ask the question, but they have no effect on TMPRSS2 processing. And we needed another inhibitor, which is known as camostat. And so we went ahead and did the live virus. We obtained a live virus from a patient in Montreal who had the D614G mutation. We isolated the virus, amplified it, and asked the question whether we can infect lung cells, Calu3 cells, with this virus, and whether the inhibitors do anything. And what we found is that the inhibitor of furin inhibit around 30-fold, diminish 30-fold the viral titer. And whereas the combination of the furin and the camostat of the TMPRSS2, we can now inhibit almost 99% the infectivity of the virus, or about 900-fold. So clearly you need both of them. The viruses uses both of them to enter lungs. Things get complicated. So this is known as the Phytogenic Pathway, whereby furin and TMPRSS2 are major players here at the cell surface. But it turns out that this virus is promiscuous. It enters in another way called the endosomal pathway. It doesn't need any of these two enzymes. And in endosomes, it actually uses catepsinel to activate itself. So it gets a very complex phenomenon. You get those two enzymes that we identified, but there is another pathway that the virus will use to get in. Now, if you look at the way this virus is spread around the world, you can see that it's really infecting almost every country. Brazil is still suffering a lot right now, as it is. This is the latest data. France has a lot of this virus, as you can see. So does Sweden, and so on. And so, but you can look at it, it now as we're entering the fourth wave. It's coming up. Look at the curve. More than 130 million died. Are infected, and 2.8 million have died. So what's the solution? Will an antiviral work? Maybe, but I think the vaccine is the most promising one, and this is the number of countries that have been vaccinated. You can see Africa is still suffering. Morocco is doing well, but Africa is suffering and has not been vaccinated. The worrisome are what is known as the variants. Okay, what we heard about the UK variant, it turned out that it mutates a site just close to the furin-like site, as well as the D614G that I told you, the patient that we isolated, and another one that enhances the interaction with the ACE2 receptor. The South African one is also a worrisome one. So is the Japanese and Brazil one. And now a California one that appeared, which affects also this. But all of them have the D614G, as you can see. And we found out, what does the D614G do? Well, what it does, it loses the N-glycosylations to amino acids afterwards. So already that changes the conformation of the S1 subunit. And the P61H apparently affects the ability of furin to cleave at the S1, S2 site. Now, what is really worrisome is the transmission of this virus to other animals. It turned out that these variants now can infect mice, can infect rodents, whereas the wild type, the Wuhan virus, did not. And now more and more we begin to see various variants that appear because of the infection in animals. Okay, you have heard about maybe in Denmark, the mink that have been infected, and the mink that have been infected created a new mutant, as you can see. And we don't know what the future is going to tell us, but we are getting mutants now, not only because of us, or because of the drugs we give to humans, but also because of the transmission to various animals. Let me now move to the cancer part. In the cancer, I think Majid was one of the first in our lab to try to identify a role of the proprotein convertases and you can see here metastasis to the liver and the growth of the tumor. This is a colon carcinoma, human colon carcinoma, HT29 and CT26. If you inject it, you can see that they colonize the liver and make tumors. But as if you put it in cells that have an alpha-1 PDX, which is an alpha-1 antitrypsin derivative, inhibiting the convertases, you get much less tumors, and they're metastasized much less. That was the first indication. He moved on many years later, as you can see, 18 years later. Now he is now in Bordeaux, and he has recently identified that, yes, it turns out that the same cells that he was using, the HT21 and CT26, colon carcinoma cells, are affected by the presence of inhibitors of the convertases. And he pointed out that one of the players here is Notch, that we had identified to be a processing enzyme processed by furin. And indeed, the Notch creates an intracellular domain, which now activates, which apparently diminishes the amount of, I mean, if you block this thing, you diminish the amount of PD-1, and hence you increase the ability of T cells to attack tumor cells. But it was not enough. It didn't explain everything. There was some other, probably other proteins or substrates that furin could activate. So one of the idea, of course, is many years ago, we had identified that TGF-beta is a substrate of both PC7 and furin. So what is TGF-beta? It's a secretory protein. It's soluble. It has a pro-domain, and it has a C-terminal disulfide bridged, if you want, TGF-beta dimer. Once it's cleaved by furin, it remains as a lasso form. It's still inactive. It's called the latent associated peptide. And it needs another protein called GERD, okay, which is in the ER, which will then combine with this cleavage protein and makes a disulfide bridge with it and bring it to the cell surface. And once it brings it to the cell surface, integrin alpha V beta 8 will interact with the RGD of the lab, allows conformational change and release of TGF-beta. So this is what, again, Majid and John Creamers just published, showed that indeed TGF-beta is a major player in regulating the Tregs, which affect the CD8 positive cells, which will then regulate the cytokines such as interferon gamma and TNF-alpha and allow for apoptosis. So an inhibition of furin will increase interferon gamma and interferon alpha and decrease metastasis. However, again, it was not enough. If you inhibit furin genetically, there are still tumors there and there are still some metastasis. So we began to ask the question whether there is a redundancy now between another enzyme called PC7 and furin. And if you look at actually an old observation was that if you look at the expression of PC7 in tumors, it's always very high. Furin changes, but really the expression of both is very high when there is a high invasive tumors. So maybe the two of them collaborate to enhance invasiveness. So we asked that question and said, how does it do that? Do they actually cleave the same substrate or do they have different substrates? So we began an unprogrammed proteomic approach, whereby the idea was that the only substrate of PC7 was the transferrin receptor, which is shed to release a soluble form. So we did a HUH7 cell, which are liver-derived cells, and we've transfected them with furin or PC7, and asked the question, what are the substrates? And we did a quantitative proteomics with Daniel Figes in Ottawa to try to find out what are the new substrates generated from PC7 or furin. And we came up with two novel type 2 transmembrane Golgi proteins. One is known as, they're really not well known at all. One is known as Casc4, as you can see, and it is shed by furin and PC7. And another, and it has to do with breast cancer. And another one is known as GPP130 or Golgi, and it is also shed by furin and PC7. So we went ahead and did all the cell biology part and showed that indeed the shedding, in other words, Casc4 changes the intracellular matrix of the cell. You can see it's completely changed from the wild type. But if you cleave it, it now expresses what is known as podosome-like rosettes, as you can see them here. And these podosome-like rosettes are reminiscent of metastasis. Basically, the cells elongate and they tend to become metastatic. So we think that the generation of the N-terminal domain enters the nucleus and transactivate a number of genes to cause metastasis. And so Casc4 itself is protective in breast cancer metastasis. This is triple negative breast cancer cell line, but it's transformed into malignant form if you have PC7 and furin. So obviously an inhibitor of both of them would be very advantageous, at least in breast cancer. If you look at the meta studies, they showed that whenever you have a high Cas4 and high PC7, your overall survival is much lower. Whereas when you have high Cas4 but low PC7, you survive much better. And furin did not do much. It looks like it's mostly PC7 here. And again, if you look at the location of the chromosome 11 where PC7 is, it is a connection to the breast cancer as well as ovarian cancer and uterine cancer. Now, interestingly, PC7 is very close in its locus to a series of genes involved in triglyceride metabolism. And the GPP130, it's amazing, this one. It turns out that amplification of this gene is very, very high in lung cancer. Look at that, 35% of lung cancers have amplification of GPP130, much more than the other genes that we know, even the ovarian cancer. So of course, the future would then be probably to inhibit these two enzymes in this kind of cancer. But then it began to tell us, what is PC7 doing? And now we opened a new chapter. Because we are interested in cardiovascular disease in my institute, there are a number of risk factors in cardiovascular diseases, which are inflammation, triglyceride, LDL cholesterol, and others, blood pressure, and so on. And so the players in LDL cholesterol, and we talk about it, is PCSK9, ApoB, and LDLR, LDL receptor. And in triglyceride is a lipoprotein lipase and ApoA5. And so what are the functions of PC7 is there a function of PC7 in cardiovascular diseases? And I will show you that these two enzymes, PC7 and PCSK9, have non-enzymatic function. Whereas PCSK9 is only non-enzymatic, PC7 has both enzymatic and non-enzymatic. And the clue came like this. We did a genome-wide study on a SNP here, which is a gain-of-function SNP. We found that in the people who have this gain-of-function SNP, they have an increased levels of triglycerides, very significant, 10 to the minus 119. That's very significant. More PC7, more triglycerides. How does it do that? Well, I was confused, of course. We didn't know how to explain this. To go right, left, we didn't really know. So we needed clues. So the clues was the tissue expression. It's widely expressed. It's in liver, adipocyte, and small intestine for PC7. And also that is on chromosome 11q23, which is a cluster of genes implicated in lipoprotein metabolism. I told you it's about 60 kilobases away from these four genes. And so Linus Pauling said, you know, one best way to have a really good idea is to have lots of ideas. 99% of your ideas are going to be wrong, but maybe 1% will work out. And that's what happened with PC7. So if you look at triglycerides, they're regulated by two ways. One way is lipoprotein lipase to transform it into free fatty acid. And they can be inhibited by ApoC3, angiopoietin like 3, 4, 5, and 8. And people use in the clinic right now monoclonal antibodies against angiopoietin like 3 or ApoC3 antisense oligonucleotide. However, nobody touched the other side, which is the activation of lipoprotein lipase. And ApoA5 is one of the activators. The other very important player in triglyceride metabolism is ApoB, because it's the only protein in VLDL, very low density lipoprotein, that get out of the cell. And I'm going to show you just in 2 words that the loss of function of PC7, we use the knockout mice, enhances ApoA5, which means decreases triglyceride because of increased activity of lipoprotein lipase, but it also decreases ApoB. That's why it diminishes the amount of VLDL. And so we just published this, and we showed that the way it does it for the ApoA5 is that it binds PC7, binds ApoA5 in the endoplasmic reticulum already, okay, and controls, if you want, the cytosolic lipid droplets and degrades it by an autophagolysosome in a completely enzymatic activity independent pathway. That was the first evidence that PC7 could have an enzymatic, a function non-enzymatic. The second came from, really inspired from the work of Henry Ginsberg in New York, who showed that if you have people who have a lot of no ApoB, and there are patients who have very low levels of ApoB. ApoB is important to get the fat from the ER out of the cell in the form of VLDL. If you have no ApoB, well, the fat accumulates, the lipid droplets accumulates in the ER. That creates an ER stress. And so the cells try to get rid of the excess fat by causing autophagy, as you can see. And that autophagy will then create a lot of free fatty acid in the cytosol. So how does the cell get rid of that? Well, it increases what is known as beta oxidation, or what you do when you take a keto diet. You force your cells to enhance beta oxidation. Well, it turns out that in the knockout of PC7 that we created, we have 50% less ApoB, which means less exit of the VLDL from the ER and hence enhanced beta oxidation. To prove that, we had to actually do a CRISPR analysis of a human primary hepatocytes. You can see here, these are IHH cells. They make lipid droplets, you can see them. If you remove PC7, look at that, you've lost the lipid droplets. So already this is a clear cut that PC7 is implicated in the regulation of fat, probably from the ER. This is an important application because in our societies, 25% of the population in industrialized countries have fatty liver, and especially if they are diabetic or obese. And this fatty liver can develop in what is known as NASH, or cirrhotic liver, or hepatocellular carcinoma. And the last two sites are irreversible. So we need to block this disease before it reaches the irreversible pathway, which is the cirrhotic liver. And there's no approved product really so far. So we believe that we are doing that. Can we actually block PC7 and diminish the fatty liver? To prove that, we needed to go into a very long experiment. It's good we had the COVID because we could do it. These are very long experiments where we gave our mice a high sugar, high fat diet for 8 weeks versus normal diet. And then after 8 weeks, 12 weeks here, we stopped giving them this bad diet and we gave them a normal diet and see how they recover. Do they recover a healthy liver in the absence of PC7 versus in the presence? That's what we got. We found that in the absence of PC7, in the recovery period, the PC7 knockout mice have a much faster recovery of the body weight. So that was good news. It means they get rid probably of their fat very fast, much better than the wild type. And the proof of the pudding is in the picture, and you can see it here. These are the lipid droplets coloration in the liver of normal mice in the recovery period, whereas this is in the recovery period of the wild type, of the knockout mice. Clearly, PC7 knockout allows burning of this fat, and you can see it here in the NASH diet. The amount of mean area of the lipid droplet is way lower. Okay, so that means that PC7 is indeed involved in the burning of the fat and may be used in the treatments. So PC7 exhibits both the protease-dependent and independent activities. It enhances ApoA5 degradation and facilitates ApoB decrease, as well as its silencing could reduce steatosis and what we think also is fibrosis because there is reduced TGF-beta. Let me finish with PCSK9. When we discovered PCSK9 in 2002, it was for us another convertase of the family, the last member. And we realized that this one autocleaves itself like all others in the ER, but not at the basic sites. It's the glutamine 152, as you can see. But it remains attached to its prodomain. And that was the surprise, that was secreted into the media with its prodomain, whereas all other convertases get rid of their prodomain. That is why PCSK9 is never enzymatically active, because the prodomain is inhibitory. Not only that, we observed that there was a second band just after the PCSK9 position, and we figured out that it had actually cleaved by furin, and we identified the site. So furin may be a negative regulator of PCSK9 activity. And so in other words, different from the other convertases, PCSK9 remains attached with its prodomain, never get rid of it, and it's actually inhibited by furin, by cleavage. So what does it do? And so I remember our old friend, Christian de Duve, who discovered the lysosomes. He said, if chance offers you a clue, follow the trail. You may not discover what you were looking for, but what you may discover may be much more interesting. And he was right. This is how he discovered lysosome by mistake. by using the wrong centrifugal force. Same thing with us. When we discovered PCSK9, first thing we asked, where is it expressed? And we're using in situ hybridization here, and we see that liver, it doesn't take a PhD to realize that the liver is a major source of PCSK9. And it was on chromosome 1p32. Now, liver is known to regulate cholesterol. And as we can see that a number of patients that we see in our clinic that have what is known as xanthoma or xanthelasma, which are really cholesterol deposits. And even Leonardo da Vinci noticed it, but not realizing that it was a disease in Mona Lisa when he painted her in the 16th century, the 15th, 16th century. And then she died, actually, that person who added it, by a heart attack at the age of 37. So she had cardiovascular diseases. And so the connection was that I would look for somebody in the world who would be interested in chromosome 1p32 and somehow in cholesterol. Luckily, I fell on Catherine Ballot and Marianne Abifadel from Lebanon, that she was doing her PhD.